Hey everybody, it's Professor Davis here, and today I'm going to talk to you about one of the largest organic molecules you may have ever seen, and that is a protein. Proteins can get extremely large. The one I've got on the screen here is actually, in the scheme of things, fairly small, but it's also really important. This is a potassium channel protein, and they were first characterized by Dr. Roderick McKinnon's lab in the late 1990s, and they actually won him a Nobel Prize shortly thereafter. This one's from a bacterium called Streptomyces. Now what it does is it allows potassium ions to traverse the cell wall of the bacterium. So the outside of the cell would be this region up here above the protein, and the interior of the cell would be in this region down here below it. And you'll notice that the regions here, which would constitute what's in contact with the cell wall, contain a lot of hydrophobic groups. These teal-colored atoms here are carbons. So we're looking at a very low polarity region of the protein here. If I rotate it slightly, you can sort of see that no matter how I orient it, there's always a bunch of these carbon atoms out here on the edge presenting a hydrophobic face. Yet at the top and bottom we see a lot more exposed oxygens and nitrogens and polar bonds. So that's where the solvent and the cell's cytosol would be. Now let me turn this thing on its side here so that you're looking at it as though you were outside of a cell looking in. Alright, now when I do that, you should immediately notice something. This protein creates a channel. It opens up a pore in the cell, which allows ions to move in. But the real question that Professor McKinnon had was, why is it that this protein transmits potassium so well, but sodium so poorly? Because potassium ions are larger than sodium ions, so if it's just a question of size, the sodium ion should move through better. But in fact, this particular protein transports about 10,000 potassium ions for every one sodium ion that gets through. Now, the key to solving this was for him to crystallize and characterize the structure of this protein in the presence of potassium ions, and that's exactly what he did. And in fact, this structure contains, which I haven't shown you yet, a potassium ion inside of the pore. So we can see that the potassium ion inside the pore is in contact with all these red atoms. And of course, if you remember from your CPK coloring scheme, those red atoms right there are all oxygens. And oxygen is very electronegative. So this is a pore which is filled with the negative ends of dipoles in the bonds that are forming it. So what does that mean for potassium? Well, that means that the positively charged potassium ion can interact favorably with the negative ends of the dipoles of all these carbon-oxygen bonds pointed inward. But sodium, on the other hand, it's too small to make contact with every single one of those atoms within the wall of the pore. So sodium would rather stay in solution where it's hydrated, interacting with water dipoles instead of those presented by the protein. So let me take this potassium ion out and show you what it's like to be a potassium ion on your way into a cell. I'm going to grab my protein structure and we're going to fly through that pore. So here we are, a sodium ion, or a potassium ion rather, moving into the pore. And all we see are red oxygen atoms. All we see are the negative ends of dipoles, stabilizing us as we move into a water-filled cavity in the center of the protein. At this point, if you look through the cavity, you'll see even more stabilizing carbon-oxygen bonds pointed with the negative dipole inward. So this is the force that stabilizes the potassium ion as it moves through the channel, and the secret to the selectivity of these potassium ion channels. And that is what won Dr. McKinnon his Nobel Prize, and that's the topic of today's micro lecture. I hope you enjoyed it. I'm Professor Davis from ChemSurvival.com and the YouTube channel ChemSurvival. I'll see you next time.